I would think you might have had things like all those things. For some of you, for some of the like the MCAT students, endoderm, mesoderm, ectoderm, those kinds of which I'm sure you had at various times in developmental physiology. But you look at certain systems and how they develop, certain things like that, and that's apropos of me talking about. I mean, because he's ready to walk pretty darn soon. So, between my wife and I, as you may or may not know, age. So we know the signs of potential walk is coming our way. And then we're all in big trouble. Why are they when you put so I'm racing out Saturday morning going, oh my God, I got to get outlet covers. Go now before he wakes up. It's been a while since we dealt with a toddler in bond. It's okay. But I digress. So that's where we are, so to speak. And so this is I was going to record something from Friday, but I had just so much other stuff with him coming to visit that, and I know you miss hearing my dulcet tones ever so much. So same thing. This is 26. We did this for anatomy, a very very brief review of those types of things, and again, anatomically, you remember there's the primary reproductive structures, which are eggs and which are the ovaries and the testes, rather, and everything else is an accessory structure. And we know that these things don't become active until puberty, per se. But I mean, again, that's a fundamental difference. The male system really does not become active from a physiological standpoint in sperm production until that time, where the female is more complex because the eggs develop during gestation and are suspended. I mean, those are the typical questions. What state are the eggs? You've had this stuff. Now, I'm sure. Eggs are suspended. At what state are they? Most of you guys know whether it's prophase one or metaphase one. Remember off the top of your head which one it is? I can't hear you thinking. My thing is metaphase one. But at least you're volunteering. That's why you're special. It's okay. So I've had a tough day. I'm waiting for test results. So my eldest has, and I love it. They tell me all about this stuff. He's got these, but these are the military calls them go no go. So he's got a whole thing about navigation, and he has a whole thing. And then they do all these simulations. So when they say go no goes, if you don't pass them, you're not going to your next assignment. Fortunately, his next assignment is basically going to be dealing with a very large mess that you hear about every day in the news sitting over there in the Gulf. But I digress. That was it's his mess to clean up. So let's hope we're not going to get any adverse headlines. He's a trooper. He'll do great. Okay, moving on. So we know it doesn't become active puberty. We know those things. We that last unit, you actually look at the different steps. There's a significant degree of complexity to this when we look at that. And again, when I explain the physiological aspect to you, a lot of what I understand about the system is from a couple that the system didn't work. That to understand what goes wrong so much as the what goes right. That's usually the frame I give everybody about it. And without going, we know how they bring them together. We know what fertilization and zygotes and gestation and parturition are. The commonality of it, how much I like homologous structures. So, I mean, a lot of this is go overable stuff. Things that are important and that could come back from a test perspective. It's really to understand what stimulates what. And it all comes back to this axis. So understand it's the relationship between the hypothalamus, where it senses various levels, the release of the very, very significant, the interaction of sensing low levels, particularly of estrogen and testosterone, and then levels of another hormone that plays a role significantly called inhibin. And you might remember us discussing this in passing a little bit when we discussed endocrine physiology. This is where it really comes back in the fold. So understand that, and the release of this very vital release factor from the posterior pituitary, this just the acronym everybody's familiar. We always say when it's just easier than saying gonadotropin releasing hormone is GnRH, and the GnRH targets the anterior pituitary. Remember of its important sexually oriented hormones are FSH and LH. Particularly, we get the prolactin, the old not sure, but plays a role in lactation and blah, blah, blah. Follicle stimulating. What's the follicle? That's something we'll look at it here today. More specifically, the follicular area is the area where either eggs or sperm are produced, and luteinizing hormone, which is stimulating in the male those interstitial cells of Leydig. You might remember them from anatomy, but they're basically going to make testosterone. And interestingly, at the same time, and there's a subtle variable with until we really for when puberty occurs, maybe that's the better way to say it, of those interstitial of Leydig producing that aforementioned inhibin. Okay, and so you can see all of those are play a role. The GnRH from the hypothalamus, again, based on sensing levels of inhibin, estrogen, testosterone, principally in the bloodstream, the FSH and the LH, and then and obviously these are the big players. Negative feedback on the hypothalamus, negative feedback also on the anterior pituitary. The inhibin, and this is true for both the male and the female, negative feedback on the FSH release from the anterior pituitary. So these are all playing a role, and these kind of diagrams actually I think are relatively easy to understand. I have no problem using this. So hopefully you do not either. This is the physiological nugget. Different for everybody. There's a whole lot of theories about when both men and women become sexually active. If I was going to, I don't mean to broad brush it, and it's not necessarily my area of expertise. We are seeing, I think, it coming a little bit earlier than it was before for both boys and girls, and certainly with vis-a-vis the female, where you're seeing menopause coming later. And I don't know why. Can't explain to you. But I mean, those numbers are constantly, it's like lifespan. Hey, thank goodness. That way I can't go when you're going up the aisle or down the aisle for graduation. Do you think I'd really do that? Because whoever I'm sitting next to would give me one of these. But you understand what I'm saying. So we know what puberty is. Life, when reproductive organs grow and become functional, and it's not an immediate process. Okay, so theoretically, the earliest time that is possible. And you're always going to see conflicting evidence. Typically, the general theme is girls, it's very unlikely to see pregnancy during that first menstrual cycle, and even a few cycles afterward. Boys, the evidence suggests that the possibility of actually fathering a child could come pretty darn early. So before puberty, low levels of these hormones are enough. It doesn't take much to suppress this by the hypothalamus. As it approaches, less sensitive to inhibition, and it begins to, and why that occurs then, and why we're seeing time variation, I don't think there's an explanation. There's only theories. And this so-called axis, the anterior, begins to release very increasingly. They release more sex hormone. And interestingly then, the threshold for inhibition, that's part of achieving this normal level that frankly boys achieve much, relatively speaking, more rapidly, and it lasts more or less for their lifetime. Okay, as you will see. So the threshold for inhibition continues to rise, resulting in more and more sex hormones, and then finally they get to the adult pattern. Is there, again, the time for this from point A to point B is certainly variable. That's the way I would, and we know from that I don't want to rebuild mitosis and meiosis. I don't want to go with those different things, but the important events are definitely part of this. particular unit. Like I said, there's really only two. It's all about diversity. That's all it is. Mitosis, all the same cells all the time. Meiosis, different cells. Thank goodness if I had kids, they'd all look like me. That would be terrible. Not one of you saw the humor in that. I would hate to see my daughter at 5'8.5" and over 200 pounds. So, and we know it's a reduction division. We know what sister chromatids are, and we know what homologous pairs are, and the whole crossing over thing, whatever name you want to give it, crossing over, chiasmata, it doesn't matter. Whatever name you want to use, fine with me. And then, obviously, the difference between haploid and diploid. So if I use those terms, Lord knows you should need to explain them, particularly in what gametes are and any of that, and going through the sequence of events that are there. So during meiosis one, that's really where everything kind of shakes. Okay, even though it's a reduction division, that's just because we don't have that duplication stage early on. We don't have that, and that's why there's two meiotic divisions. Okay, it's the significant events that are there. And so at prophase one, synapses, chiasmata, whatever, recombination. Anytime, all of those are terms for when genetic information switches hands. So whatever you want to, and we know the two pairs of sister chromatids are in close proximity, and the middle two strands, like little legs and arms crossing over and exchanging information. We're all familiar with that, fine and dandy. And then so all of that happens. So now we have these the two joined sister chromatids of each homologous pair are distributed at opposite ends of the cell. And then what continues to go on with regard to it here, when we get to metaphase one, now we have the second event, the law of independent assortment. And the interesting thing, to me, it's just fascinating how long before science was science, how long ago was Mendel doing this? This monk in Prague, at that time Czechoslovakia, rather than just a Czech, how could, just from looking at plants, be able to conceive of a model where all this could be explained? It's fascinating. So voila, that's what happens. And after that, the rest is history. So you can look through all of those as you wish. So again, the function, reduction division, genetic diversity, random alignment, crossing over the two big features. We're all aware of those. I can't even read that. And I can't get through these fast enough. I don't know that you get a whole lot out of that. I'm having trouble playing them here. Yeah. Let's see.
The process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with two sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war.
I love that.
At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
And I think the take-home point again is the distinction between male and female. In female, we were looking at polar bodies, first and second polar bodies. So it's different here. We're producing 4, when in that we're producing 1. So, and we won't review the anatomy any more than is necessary because we went over that. But if you don't recall it or you just need to review it, then please do so. I think that's the only thing that's, I would say, it's important for you folks to do. The role of the glands would be important. And that's a crossover. Again, the vast majority of the ejaculant, the seminal fluid, is made of seminal fluid along the posterior bladder. And again, the pH comes into phase more when I do that last unit that I really don't test on. Okay, but again, the key ingredient, fructose, because that's the only thing that energizes sperm. Coagulating enzyme, which is important to make sure that the sperm effectively don't leak out after the point of ejaculation. Various prostaglandins play a role in any one of a variety of things that go on, both in the release and then subsequently after ejaculation, events that go on after intercourse. That's there. They classically associate with that pigment we use for identification vis-a-vis forensic matters and a large amount of the volume. And again, prostatic fluid certainly has that typically that white milky color. Again, the source of that PSA that's so important. And that may or may not be on it. I'll let you know when I do the narration, again, verbally, whether that's on a place that we had in our orbit. A friend of my wife's, she came in with oh my goodness, a PSA at 10. It just about fell over. I mean, it's not a happy tale. A lovely person who unfortunately life has played some cruel tricks on, but and very often, I think every family and every close-knit circle of friends has those as well. Oh, by the way, thanks a lot. Because my wife and I, we're very lucky. So unfortunately, we were cleaning out, as an aside, we were cleaning out trees and stuff and shrubs and doing some stuff last week before the rain's coming. My wife has got a dose of poison ivy. Yuck. So I was wearing pants and she's wearing shorts. Oh. Front row. Tactical error. You don't want that stuff. And she breaks out all over the place. trying to get her to go to urgent care. Well, I'm busy. To get the heck over there. So I was going to say something else. And again, I think that's important. Not much else to say about any of these things. The prostatic disease, we covered that last time, etc. The cancer, the nature of what semen are, and things like that, characteristic. Which brings us more to this. I don't really delve a lot into these sexual responses and things like this. This is important. I'm sure there'll be aspects of this that'll be on the exam. It's, I first have to understand, but mostly we look at arterioles. We know they control blood flow, but these are usually constricted. And there's any number of things that causes them to be stimulated, whether they're mechanical, whether they're things that are mental or emotional, whatever these things might be, certain signs and signals that they discussed here. But the CNS activation is by way of, and this is important because we looked at this in endocrine, and we looked at this in the autonomic nerve. This is the ultimate autonomic dualism in response, you might recall. Parasympathetic nerves innervating, as they're called, the internal pudendal arteries. That you don't have to know. And it releases that interesting, which we did not know about, which is nitric oxide. And that nitric oxide, the amount that is produced in that particular area, causes those arterioles to dilate, and we have the inflow of arterial blood. Okay, the arterioles dilate, those corpora, the cavernosa and the spongiosum, expand sufficiently because of the amount that's released in that particular locale. And what that happens, it precludes venous drainage. So the end result is the engorgement of the erectile tissue, enlargement and stiffening of the penis. It's a very well understood process. Remember, the spongiosum was the one that surrounded the urethra, so it maintains it to be open. And there's all sorts of things without going into whatever, and everybody's different. I think we're all aware of that. And it can be induced by or inhibited by higher mental activity. So however you want to describe it, any number of factors can either make this come on more easily or make it effectively not likely to come on. Okay? At that point in time, now the sympathetic nerves, they're the ones who play the more active role in the process of ejaculation. So we get this dual, these two systems that normally are opposites working together. So it's one of those wonderful aspects of synergistic activity in two opposing systems, typically opposing systems. Spinal reflexes, and they basically stimulate. Remember that the sperm cells mature in the epididymis. That's where they effectively achieve their potential for motility, for ability to move, and then they make their way into, then effectively, as the smooth muscle contracts in that area, it starts that journey through the ductus deferens, seminal glands, prostate, all that type of stuff, and you can review that. The bladder internal sphincter muscle constricts, preventing the expulsion of urine and or reflux of the semen into the bladder. And then effectively it's ejaculated. So the bulbospongiosus muscles undergo the series, remember those are the ones surrounding the urethra, to expel the semen at a relatively good rate of speed, and that event's accompanied by the intense pleasure referred to as climax or orgasm, and the accelerated heart rate and blood pressure are all very consistent with sympathetic activity. And then the resolution, this is the time to, and it varies, I think, from individual to individual. Okay. I think in general, if you want to, again, paint with a broad brush, as you're younger, the resolution period is relatively short, and as you get older, it's considerably longer. And it doesn't always follow any of those patterns. There's so much variability. All of that. And again, the sympathetic then controls and brings it back to its original. So the so-called latent or refractory period, during which the man is unable to achieve another orgasm, and you can see the variation. Consistently lengthens with age, But that period you can see can be, for a young person, short answer, just depends. No way to predict it. So parasympathetic nerves release too little, and that should be a big capital zero, or capital rather, to be nitric oxide, leading to ability and effects, as you can see, over the age of 40. And that's the interesting thing. Before we had the availability of these ED medications, if it affects that percentage, then how we're then able to conceive with their partners' children. I mean, it begs that question. And the short answer is I don't think anybody knows, but it's interesting that I do not, it's trying to think of the right way to express it. I don't know that I think there's a lot of variability to give that hard of a number statistically. I wonder if, and I don't know if they ever sought to measure it, because really until, you familiar with like the Kinsey Institute at the University of Indiana? It's not ringing a bell. I had this discussion with my micro class. We are a very provincial nation, meaning we're relatively new. And I don't know about you guys, how you grew up or what your family members were like. I know me being a couple of generations, we didn't talk about that stuff. Our parents do. If you ask my mom and dad, it's like, no, they were hastened. Well, to anybody else. It wasn't discussed at the school. You were lucky if they got, well, maybe we can get together. And they did with my family. True story. With the guys and gals, of course, would be separate doing this. They had a bunch of us guys who were all the same age. Somebody got together and got one of the local, general practitioners to have a chat with the boys about the birds and the bees, so to speak, to try to get through with that. Not to, and to try to at least clarify all the misinformation. This is not so the case in Europe, not so the case in more in cultures that have been around for a long time. Okay. So it sort of amazes me, you know. So that's why I don't think we have a realistic way of comparing these because nobody studied them. And so really the first people to study this, it did a movie years ago with Liam Neeson, quite a few years ago, maybe 30 years ago, about Kinsey and the Kinsey Institute, a husband and wife doctor team, and they did a show on Showtime about it. Maybe 10 or 15 years about it, like a short series about what their explorations in studying human sexual responses. That was unheard of. Absolutely unheard of. People were horrified over things like this. And I mean, I think that is the reason why, and I said this at my micro class, and we talk a little bit about this when we do the female repro stuff. Why does this nation that has so wonderfully educated, has outstanding health care, outstanding doctors, why do we have such a ridiculously high rate of sexually transmitted diseases? Can you figure that out? And nobody, it's the shameful lack of education to the very young, and not the misconceptions of you can't get pregnant the first time. Oh, you can't pass along a STD the first time, and blah 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 blah. What a bunch of inadequate and incorrect information is being passed on. Now, understand, I am an authority on this because I'm hopeful that I'm not talking to anybody who has had a baby before they were 16, like my own daughter. There's a reason it was my great grandson at age 11 months who you just saw in that wonderful picture. So my daughter's a grandma at 39 actually. So you certainly are listening to an authority on this. So frame it that way. But now we understand about these kinds of things. Typically alcohol, a variety of drugs, long-term use of blood pressure medications. Antidepressants were notorious about this. That was one of the big side effects that people saw, and maybe they've corrected it today, but we talked about it a lot when they started with Prozac and those kinds of things, the SSRIs. even though they didn't really have anything to do with this system per se, and seemed to be a well-known side effect with regard to. So the cottage industry, that's erectile dysfunction, basically work on the pathway with nitric oxide. Nitric oxide is broken down by, just like you've learned about in many of your courses, a metabolic pathway. They interrupt that pathway. If you interrupt the breakdown pathway, it stays in your system longer. So the nitric oxide is not broken down at those sites. And so once someone is able to get to a state of sexual excitation, they can maintain it. The pitfall is that sometimes people respond, and sometimes it's hard to get rid of it, and then that requires some pretty significant intervention to avoid permanent damage. Just say. So chronic, this is understandable, and I mean it's been a extraordinarily successful. So many companies make it now, and now in this day and age where online medicine, I mean, years ago you had to go to your doctor and all that type of stuff. Now anybody can, for whatever reason, go on and get prescriptions for this type of stuff, get it sent to them. Well, I don't see anything wrong with that. I really don't. I just think people have to be wary if they're experiencing side effects to not ignore them and just dismiss them to say, oh, it's just a medicine like that. That's the difference. I mean, yeah, I think you should be very cautious with regard to people who use them, or in your orbit, people who use them. So problems with blood vessels, okay, poor circulation, varicose veins in that area, all of the variety of different nervous system disorders, diabetes, a very frequent. Again, diabetes targets particularly small blood vessels because it tends to deposit in and around the basal lamina and really predisposes small blood vessels to arteriosclerosis. And so all of these, it's not that they're enhancing the effect. I think that's mistaken. It's that they're just prolonging the effect by not letting it be broken down. I mean, effectively, that's what happens. So I think that's a bit of an error in the way that they're describing. Let's see. And then, maybe the last part about this is understanding a little bit of spermatogenesis. Then we're basically done with this forming male gametes. Remember, they start in the seminiferous tubules. Again, and so this is closed tubule system, different from the female where we have the opening, where the egg is ovulated into the oviducts. And I mean, that immediately creates an area where infections can spread. Again, I'm doing that unit now in micro, basically the urinary tract, and so let's say urogenital disorders and urogenital infections that are there. As you can see, begins roughly around 14, some earlier, some later, but I'd say that's probably later than I would estimate by most. And I mean, a substantial amount are made daily that are there. The tubules, you look at the epithelium, but they have supportive cells, the spermatogenic cells, muscle cells, these myoid or muscle-like cells. They're more about moving it along. And again, these interstitial cells of Leydig, they are endocrine in nature, androgens and some estrogen. And they give you an idea of that cutaway that you saw in anatomy and a little bit more here. Basically, in the seminiferous tubules, you have what are called spermatogenic cells. And these, interesting, those stem cells in that area, they start with mitosis. So they form two types of spermatocytes that are there. You see, they follow it down a little bit later. Some that just stay there and continue to do mitosis, and the others that go on the maturation process to actually be sperm that are there. And then the ones that are going to make their pathway into sperm, basically now they start to undergo meiosis. Those spermatocytes form secondary spermatocytes, which form spermatids. So you just have to look at that logically and take a look at that pattern. And so once they're spermatids, then we enter, and the spermiogenesis, that's something that occurs for all intents and purposes in the epididymis. That's that transition from something that you would not recognize as a sperm to actually becoming a sperm. So It begins at puberty in the stem cells, direct contact with that base layer continuously. The type A remain there, and they're the pool of germ cells. The type B ones move on to become the spermatocytes. And if you can follow this, and it's hard, it's just blow these things up, it just kills me. Oh, heavens, Betsy. Here we go. So you can see the type A cells. Here's the type B ones. They become the primary spermatocytes, then secondary, and then finally, when we get into the spermatids, now these are ones, and you can see they're starting. This is where we begin to see the changes. And finally, these spermatids really undergo full maturation as they become later spermatids and finally sperm. And effectively, what they're doing in this maturation, you're developing the components, the head, the midpiece, and the tail. The tail is strictly for moving it side to side. The only place we have a flagella. It's there. It's all within. Remember, it's all membrane-bound organelles. So this is a membrane-bound appendage. This is the engine, mitochondria utilizing the fructose, and then you have the head, and the head contains the genetic material, the haplogenetic material, the dad contribution. You want to look at it that way, and then the helmet-like portion called the acrosome is all about being able to penetrate the exterior border of the egg. That's it. Nearly are mature. So, and then we go in with that, with the secondary spermatocytes, the spermatids, all the genes are turned off and DNA is compacted into those pellets. So basically they become this compact unit dedicated for that. And in terms of infertility, if you may have heard me say this before, because most of you were there for anatomy, effectively, see what's going on, we got a little bit more time, with regard to the anatomy aspect of it. And I was talking a little bit about it's not an area that I have a tremendous amount of expertise about, but about when we talk about infertility, we were told, you might remember, we went to a world-renowned expert at Hospital University of Pennsylvania. 20% male, 60% female, 20% we don't know with regard to what yields infertility. I mean, so there's always, even though we see most of the problems associated with a woman, less so with a male, maybe a 3-to-1 relationship. And then we have that unknown area, if you wanted to paint it again with a broad brush. So one of the things from a male perspective, you provide a sample. And in that sample, you're trying to determine if there's an adequate count, if there's adequate motility, if there's any malformation. You just look at, basically taking a look microscopically and see if it ticks all the boxes why it should work. Okay. And then when we were talking about in vitro and variants of in vitro fertilization, the next step, because for donor sperm, if you're going to use it, you have to freeze it, till it goes to wherever the IVF is going to be occurring. And some sperm don't freeze, and when they unfreeze them, they don't work as effectively. They lose some of that motility. So now maybe this has changed. My first wife and I were investigating this more than 45 years ago. So but the techniques were in their infancy then. So just to kind of give you an idea about that. What's going on. In the female, again, you're going to see a lot more complications in there. So, and it goes over that. The head, again, the genetic material, the acrosome, particularly that enable the penetration of the egg. As we look through it, you're going to see it. That's why there's such a vast number of sperm in the ejaculant, because it's like an onslaught to break down that boundary until one finally penetrates. It's this large action, although only one, for the most part, penetrates. Obviously, now we start to get some interesting disorders, chromosome disorders, if there's more than one penetration. You start seeing some of those variations. And the midpiece is metabolic, and the tail is just removing it around. You can see it just as easily. This is a typical micrograph. We always draw it this way, but this is maybe an easier depiction for be able to see 64 to 72 days if conditions are hospitable. They're unable to swim per se, but pressure of testicular fluid pushes the immotile sperm from the epididymis. And once there, then once they exit the epididymis and get into the relatively long, remember it's like 45 cms. So maybe you're looking at something about 18 inches, that process till they gain their motility. So the supportive cells certainly are important. The tight junctions, it's all about that barrier. It's so important. The blood testes barrier, we talked about that, the immune system. Sperm not formed, results of sperm not being recognized as self. The testicular fluid that's produced along with this by the supporting cells, rich in androgens, metabolic acids for transport. Really, the cells we have a whole mechanism for phagocytizing those that are quote unquote faulty. And you can see we have a variety of these mediators that play a role. The androgen binding protein keeps the testosterone levels high to stimulate spermatogenesis, and then the inhibin inhibits spermatogenesis by inhibiting FSH released by the anterior pituitary. So, there's a lot of things that are the control. I'm not too worried about the androgen binding protein. Depends on what you go into to look at this. This is the interesting, this was 8 years ago. And that's why I wonder about all of those things. The need for ED medications, the different ages, we're seeing different ages on earlier, later. And I don't know how confident you are. A lot of people point to the food system with chemicals that are in there. Whenever my wife extensively reads, looks at podcasts, very interactively on these things. She's such a big proponent of that. And it's there. So, I think you have to look closely. She was telling me about a way that there's a scanning code if they don't put all of the ingredients on a consumable. And it's like a QR code that you can scan. Look, you're young. Stuff you should be doing. We're older. We're concerned about it because we'd like to stick around a little bit longer. So here, all of these things, problems with sperm quality or quantity, gradual decline in male fertility. That's interesting. That's where that number is so high. So it makes me, the very fact, they're talking about erectile dysfunction. The question is, does that mean men are going to be more productive? I don't know. I don't think that's ever been really studied elaborately. Foreign molecules, toxins, plastics, pesticides. Oh my goodness. It's a night field. Other estrogen-like compounds, antibiotics, all these types of things. We know about excessive alcohol. Marijuana is interesting in that we really can't quantify all the effects of, and your choice. If you're a gummy fan, go for it. Maybe that was it for me smoking marijuana daily for 7 years. I always felt it enhanced my understanding of things, which is easy to lie to yourself. Who knows? Maybe that played a role, or any of these other things. Okay, and obviously the temperature one we can acknowledge as being there. Here's that axis again. As you should be, at least we've already gone over. Again, the difference between follicle stimulating and luteinizing is important. That follicle stimulating indirectly by stimulating cystenocytes to release this, and they keep the concentration of testosterone high. So it's effectively you're playing an important role in making sure it's available. And you can look through this as well as I can. Rising testosterone levels, triggering that. Testosterone enters the blood, stimulates maturation. We know about that. The secondary sexual characteristics. that are there, etc. So, interesting. Immediately before birth, testosterone levels are there, and then they're gone. And I guess that's part of a developmental process. I do not know. Okay, and then as puberty nears, then all of a sudden those levels have to reach sufficiently high to suppress that. So you can see fertilization. Look in the gestational stages, even at birth, then it goes off, and then more or less, pretty consistently, we see this. I mean, once they reach, let's say mid-teens, stays high, and it begins to. I mean, it doesn't mean there's not any production, but the production diminishes. And a lot of people have sort of anecdotally talked about maybe if you want to think of it as quality, maybe there's an inherent risk of some problems associated with octogenarians, but there's ample evidence of them being able to, sire a child at that age. Okay, so it takes about 3 years to achieve that balance where they're fairly stable throughout life. And if the GnRH and the gonadotropins weren't around, it wouldn't work. So I think that there's a little bit more, but I'll save it, and I'll just finish this up next time on Wednesday, and we'll get into the female aspect. Thank you. And we'll head over to the chairs are more comfy over in the environmental science.